In Chapter 18, we will describe the structure of proteins, the molecules that carry out the majority of the biological processes essential to life. Proteins are polymers of alpha amino acids. As the name suggests, amino acids have two essential functional groups, an amino group and a carboxyl group. Typically, amino acids have the following structure, the one that I have shown right here. But oftentimes, if you look in other books, sometimes you'll see them written like this, where we have the amino group, the alpha carbon, and we have a carboxyl group like this. Okay? But you know that a carboxylic acid is acidic, and an amine is a base. Okay, it's an organic base. So what happens is the carboxyl group is going to donate this proton, and the amine is going to accept it, and what you end up with is this structure over here, where we have the nitrogen with an extra hydrogen so that it has a positive charge, and the carboxyl will have lost the proton to become a carboxylate. Overall, we still have a neutral molecule, but this is more correct, showing this ionized form. Now the carbon that has the carboxyl group attached to it, this is what we call the alpha carbon, and you can see that the nitrogen is attached to that alpha carbon, and that's why we call these alpha amino acids. The natural amino acids all have unique R groups. When the R group is simply, if this is simply a hydrogen, this is the simplest amino acid, and it's called glycine. The amide bond that forms between the carboxyl group of one amino acid and the amino group of another is called the peptide bond. That's this bond right here. The joining of amino acids by peptide bonds produces small peptides and larger proteins. Because protein structure and function are essential for life processes, it is fortunate indeed that the amide bonds that hold them together are not easily hydrolyzed at physiological pH and temperature. The process of protein synthesis in the cell mimics amide formation in the laboratory. It involves acyl group transfer. There are several important differences between the chemistry in the laboratory and the chemistry in the cell. During protein synthesis, the amino acyl group of the amino acid is transferred, rather than the acyl group of a carboxylic acid. In addition, the amino acyl group is not transferred from a carboxylic acid derivative. It is transferred from a special carrier molecule called a transfer RNA, or tRNA. When the amino acyl group is covalently bonded to a tRNA, the resulting structure is called an amino acyl tRNA, shown here. The amino acyl group of the amino acyl tRNA is transferred to the amino group nitrogen to form a peptide bond. The transfer RNA is recycled by binding to another of the same kind of amino acyl group. More than 100 kinds of proteins, nucleotides, and RNA molecules participate in the incredibly intricate process of protein synthesis. In Chapter 18, we will study protein structure and learn about the many functions of proteins in the life of the cell.